Hello, people. Now we will be learning about the opportunistic mycosis. It includes candidiasis, cryptococcus, aspergillosis, and mucormycosis. So, firstly, we will learn the candida. So, candida is yeast-like. So, in this, the candida albicans. Candida albicans show two phenomena. First is Renault broad phenomena, and second, chlamydospore formation on cornmeal agar. Now, the Renault broad phenomena, the colony's growth plus human plasma shows germ tube formation, which is germ tube test. Test is done to differentiate Candida albicans from other species. And the chlamydospore formation on cornmeal agar. So, on cornmeal agar, which is the nutrient deficient medium, chlamydospores are formed at 20 degrees Celsius. Now, the non-Candida albicans are Candida cruciae, Kefir, Glabretta, Stelatoida, Guillermondi, Tropicalis, Vishwanthi, and Aureus. So, these are Candida cruciae, Kefir, Candida Vishwanthi, and Candida aureus. These are non-Candida albicans. Here, we can see the germ tube in the this is germ tube, which is Renault broad phenomena, and here the Candida albicans is showing the germ tube, and here this is the chlamydospore, which is formed on the cornmeal agar. Now, the Candida albicans diseases. So it is oral thrush most commonly. So second is esophageal candidiasis, third vaginal candidiasis or vulvovaginitis. Candidemia means Candida yeast cells in blood, emia. And the candiduria, Candida yeast cells in urine. Onychomycosis, nail folds are involved. Onco, nails. 7. The urinary tract infection and the upper respiratory tract infections. And meningitis, which is rarely involved. Now, the Candida infections are common in neutropenic individuals, or we can say that the individuals who are having the less number of neutrophils. CMI, the person having the decreased CMI or cell mediated immunity. For example, in AIDS, malignancies, the CMC, chronic mucocutaneous candidiasis. Now, the lab diagnosis of Candida is tissue biopsy and samples. Then, the gram stain, budding yeast cells are seen, and colonies cultured on STA appear creamy, pasty, smooth. So, colonies cultured on STA, they appear creamy, pasty, and smooth. Sugar, carbohydrate, fermentation, and sugar assimilation test, which are used in identification of Candida albicans and other species of Candida. So, third is germ tube test, and fourth, chrome agar method. In this, the colorful agar is used for identification of Candida albicans or the non-Candida albicans. ELISA for antigen detection. So, tissue lab diagnosis is tissue biopsy. The gram staining, budding yeast cells are seen, colonies may appear creamy, pasty, smooth. Sugar or carbohydrate fermentation or sugar assimilation test, germ tube test, chrome agar method, and ELISA for antigen detection. Now, here we can see the growth of Candida albicans on SDA. So we know in SDA we can see the creamy, pasty. Here we can see creamy, pasty, and smooth colonies are seen of Candida albicans. Now, let's see the gram stains smear of Candida albicans. So it is budding yeast cells are seen,